Hello, welcome to this May 2016 Get Started with CyVerse webinar. I'm Jason Williams coming to you from Cold Spring Harbor, New York. Welcome. This is a monthly webinar that we hold for all of our new account holders with CyVerse. So every month, approximately 600 or 700 new users join us. And this is an opportunity over the next hour to give you a chance to understand a little bit about the project and, of course, about the tools and resources that we have available to you to help accelerate your research and education. Before I actually go into the presentation, I want to familiarize you with a couple of the features of this Adobe Connect room that we're using in order to broadcast the webinar. First, importantly, if you look at the bottom of your screen, you're going to see a place where you can actually enter your questions. And off to the side here, I can go through and see whatever questions people have left for me. So please, at any time, if you have questions, go ahead and post those at the bottom there. Another important feature in the room is off on the side. If you look, there are some important links and downloads, so you can take advantage of those. And at the very top of the screen, you're going to also see like a little box with 4 arrows in them that you can actually make the video full screen. And so when I do some of the demos from my computer, you can actually see in a little bit more detail. Another important thing that is going to help you is that we're going to record the entire thing so that if you've missed anything over the course of the next day or so when that's posted, you can go back to YouTube and see it. Now, I do have one and only one user reporting that they can't hear me. As far as our studio here, everything seems fine, so I'd suggest that we can ask that user to go ahead and leave the room and come back. Chanhua, could you actually chat that user if you double click on the participants and just suggest to her that maybe we can, I think you have to do it from the other side where it says participants, and she should be in the list there. And you can send a chat message to her, I think, or in the chat. So if not, maybe she'll figure that out. Okay. Thank you. But for anyone else, if you do have any type of audio-visual problems, please, let us know. Or in the rare case where that might happen, you can usually just leave the room and come back and it will be solved. Okay. So let's get started. The outline for the presentation is I'm going to give you about 10 minutes or so of being able to get an idea of the history of the project. After that, I have several demos that we're going to take you through. The first one is actually how to upload and manage your data. So uploading the data, sharing those data, searching for data sets, we'll cover that. The next thing will be our discovery environment, which is a web interface to the bioinformatics applications. It also has some features for data management. We'll see those. And then finally, we'll do a live demo of also the cloud, which is Atmosphere. It's a technology for making use of biological applications within the cloud. If you've never done that before, we'll go through what all of that means. So why don't we get started with the presentation, which is just a quick introduction to the project itself. Okay, so for the actual project, there is a vision and there's a mission that's actually associated with CyVerse, and these things are important to us because they really shape the direction that we'd like to go in. As far as our vision for the project, it's to transform science into what we call data-driven discovery. So it's the ability for scientists to be able to use and access data sets, and then they will be able to actually derive information from those data sets and be able to discover through the actual just the use of data. That whole idea of data-driven discovery, data science, is really enriched by the quality and the quantity of data that we're able as biologists to obtain from sequencing, from images. So really this whole idea of data-driven discovery is the vision for CyVerse. Now the way that we actually do that or accomplish that vision is by delivering a national scale cyber infrastructure for life science research and by training scientists to enable them to use it. So we're more than 30,000 users. We're petabyte plus of data, hundreds of publications, courses, discoveries. So it's really as a community that's devoted to using these tools and accomplishing that vision. Now, as many of you may be familiar, there is an actual evolution of the project. Starting back in 2008, the project started as iPlant, or the iPlant Collaborative, and its goal was to quote-unquote empower a new plant biology. Back in 2013, after having accomplished a lot of that successfully, we were actually renewed by NSF, and in the context of that, we became really a cyber infrastructure for life science. By 2016, and really that's just a few months ago, that the beginning, this renaming is what we actually were able to accomplish, this rebranding of CyVerse into a platform in and of itself. So this is the evolution that the project has gone along, and you will be able to actually sort of see that even though some of the objectives of the projects, have sort of morphed, really our core mission or our core idea has remained the same. So the other element of CyVerse that's really important for our users is to understand that we are actually a National Science Foundation project. We're funded entirely by the National Foundation. And it's something that means that we're your colleagues, we're your collaborators, and it's really $100 million of investment that's on the table that you can use in order to take advantage. So there's so many tools that are there that you can actually get access to without any cost. The entire project resources are freely available to the community, and as you'll see in some later slides, you have access to this across, of course, the US, but also we're in favor of international collaboration. So for those who are working with CyVerse in many different countries, you can actually access it and use it there as well. Finally, of course, we like when you cite us, so please visit that webpage for information on when you make use of CyVerse, how to be able to give credit and recognize everyone in the project. Another thing that I'd like to talk about is what is cyber infrastructure. So I've used that word, and I want to just give a really quick definition for anybody who's wondering. In one sense, it's not anything you haven't seen or used somewhere else in life. It's data storage, software, maybe high-performance computing is something that folks have not necessarily used as a routine part of their analysis, and also people. And that really means things like training, help, support. But it's all of these things combined and really organized into a system that's custom for biologists and that really allows you to solve problems of size and scope that you really wouldn't be able to do otherwise. So practically speaking, you have access within CyVerse to platforms, tools, data sets, storage and compute, and then this is an example, training and support. So these are all elements of what is involved in CyVerse. Another important thing about CyVerse is that we support all domains of life. So that's another element of the project that was really important to us early on. The name branding itself reflects the idea that we have a project that's built for data. So you can come to CyVerse with plant, microbes, animals, biomedical data, ecological data, climate. All of those elements are involved in being able to access and use and leverage the tools that CyVerse have meant. So if you're working in any of the domains of life science, it's really a tool that's for you and it's built for data. The product stack of CyVerse, and this is sort of a high-level view, is depending on how you'd like to use it, varies from ease of use to flexibility, whether you're at the top of the scale or the bottom of the scale. On the top level, we have what we'd call ready-to-use platforms, and this is what we're going to give demos on. So Atmosphere, Discovery Environment, DNA Subway, these are all tools that we'll talk about that are user interfaces. But if you want to be more flexible, and if you actually have the expertise, really anyone who'd like to make full use of things, the high-performance compute, the cloud system, the storage, those are foundational capabilities that are available through the API. So there really is an entire stack of resources and tools to suit whether you are a bench biologist or whether you're sort of a bioinformatics engineer someone who's at the computing systems level, you can really take the elements of CyVerse that you want to take and then customize them. For me, for most of us, we are sort of on the bench biology side, and so you can think about using and accessing those as I'm going to show you in the demos that we're about to get into. A last little portion of this sort of background history. Again, the importance here is just to understand a little bit about where we came from and why we're going where we are. That CyVerse, how was it built? Well, very early on we realized that the community was going to be super important. So getting feedback over the course of a year and a half shaped our priorities. In the beginning, you can imagine that those priorities, genomic data, analyzing genomic data, tools for assembly, RNA-seq, ChIP-seq, all of those things were really, really important several years ago when we started, and in fact, they're still the big drivers of big data within biology. If you go to our website, and this is just sort of a grab from there, you'll see that we're really concerned, and this is a difference for CyVerse, I think, than some other projects. We're really concerned with many, many different types of data across many biological disciplines. So it's not just about sequence data, even though that's perhaps the biggest driver of things, but environmental data, phenotypes, all of those things are needed in order to build higher level associations, models, analyses. We're also big on collaboration. We're not the only solution to every problem that's out there. So we work with many different projects, including some of the ones listed here. And we ourselves are an entirely virtual organization. So the University of Arizona, University of Texas, Austin at Texas Advanced Computing Center, Cold Spring Harbor Laboratory, UNC Wilmington. Those are the main partner institutions that make up CyVerse. And we're also international, increasingly so. So this CyVerse UK is the first and sort of stellar example of a group that wanted to replicate some of those resources, but leverage other things that were built and use their own funding through BBSRC in the UK to create their own CyVerse UK, as it's now called. Okay, so now we're going to get into some of the details, and let me do that by just drawing a little bit of an analogy. We think of ourselves as almost the building blocks, or the Lego blocks. So what we've built and what I'll present in the demos is this piece, that piece, and many times we really put 85, 95% of what you want together. So you can come and find the end-to-end workflows that you need, the bioinformatics tools that you need, the resources for moving those data, sharing those data. And then there are a lot of other custom things that only you want or are appropriate to the type of thing that you're doing. And so you'll also be able to go and sort of put those pieces together. So that analogy will help you as you think there are many pieces, and that doesn't mean that you have to build everything using all of those pieces. It may be overwhelming if you don't think of it like that. Okay, so here are some concrete pieces. The data store is where I think a lot of things begin. Every user has an allocation of up to 100 gigabytes. If you need more than that, that's absolutely possible. You just need to request a little bit of justification and you can get that granted. The data are automatically backed up, and that's part of your data management plan. If you look, it's quite secure, it's redundant, and it's in such a way that we've never lost any data, so that's good. And we've made it fairly easy to upload, download, and share, and I'm going to be showing that in the first few demos. I'll also cover in a demo the discovery environment, which is our interface to hundreds of bioinformatics applications. With the discovery environment, you can run almost any bioinformatics application. It's integrated with our data and our HPC, and it's also user extensible, so if you don't see an application that you like there, you can add your own. Atmosphere is our cloud computing resource, so you can actually access hundreds of virtual machine images. If you are not familiar with cloud computing or some of the terminology, I'll walk you through that as we get towards that portion of the demo. It's also integrated with our, again, computing and data resources. The APIs, such as Agave, are there for those who are going to make use at the custom level. Any of the resources and tools that we provide, you can actually customize that through Agave. I won't be talking about the API any further than that, and in these next few I won't be because they're not part of the demo, but they're very important for many to know about. DNA Subway is one of them, so these are a set of educational workflows for working with genomics, looking at several bioinformatics tools that have been put into these easy to use, I would call them even classroom-friendly workflows. It's really useful for teaching important concepts in biology and bioinformatics, and it supports a series of inquiry-based experiments. So in other words, students are actually generating data, working with real experiments. Bisq is the image analysis and metadata management platform where you can work with hundreds of different types of categories of image files or image file formats. You can also integrate your applications and work with APIs to really customize how you work with your image processing workflow. So those were some of the high-level products, and you got a little bit of the history of the project as we evolved from something that was quite focused on the plant community to something that is really applicable to any type of life sciences investigation. And now what we're going to do and spend the rest of our time is on some demos. Again, the demos are just to give you a taste and idea. Then we'll show you where to go for the full documentation and help. If you miss anything, this will be recorded, so you can actually go back and catch the latest recording on YouTube when it's posted. I'll once again remind you that at the top of the screen there's the option to make the video full screen, and since I'm going to do these demos from my computer, it maybe will improve your ability to see some of the details there. Okay, so getting data into CyVerse. As I mentioned, the CyVerse data store allows you to store any type of file related to your research. The platforms within CyVerse are all connected to the same data store, so moving data is quite easy. There are many ways to automate file transfers, and it just makes it possible for you to share files with your lab members, with collaborators, and communities. There's several ways to access the data store, and that's because biologists and bioinformaticians need some flexibility there. I'm going to cover these briefly with the live demo, and you can choose what's appropriate for you. They have different functionalities, different eases of use. The discovery environment I'm going to show to you actually second, but the discovery environment is a platform. It's like a desktop, and you can actually manage your files. There's support there for uploading and downloading small files. If you want to upload really big files, I would recommend CyberDuck or iCommands, which I'm going to show. But it is great for sharing the data because you can share your data using many of the URLs. You can also share it with any other person who has a CyVerse account. CyberDuck is the first demo I'm going to take you through, and if you've used something like Dropbox, CyberDuck allows you to have a drag and drop interface to your files and folders. There's no size limit, and it's really easy to use on the desktop. And finally, if you're used to working at the command line, iCommands is perfect. It gives you full flexibility. You can script and automate transfers if you have data that's sitting on some server. It's quite quick. And so those are all different features that you'd want to take advantage if you are familiar with that. So let me get you through this demo. I am on my home computer. I'm going to go ahead. I have CyberDuck installed. I'm going to start CyberDuck, and I'm actually going to start connecting to it. And then I'll actually talk about, well, how would you start CyberDuck and how would you start getting connected to it? So this is CyberDuck. And in order to connect to it, I'm just going to double click on this bookmark. And it'll probably take, in my experience, about 30 seconds to connect. And that's only a one-time thing. In the meantime, I'm just going to come and go to my browser here and go to the CyVerse homepage. So if you go to the CyVerse.org, you'll find the CyVerse homepage. And if you scroll down to the Learning Center, you're going to find some resources, including what you'd need in order to be able to download CyberDuck. As again, you don't have to try follow along with me, other than to sort of get these concepts and ideas of, okay, here is where you should come back to. Down here in the Learning Center, I'm going to go ahead and click on this step of managing your data. And there are there's a fair amount of information there, and eventually you get down to CyberDuck. So I'm going to go ahead and click on that link there that takes me to the CyberDuck documentation. And many of the items of documentation exist on the wiki, so I'm just waiting for that to come up. Okay. And if you go through, it will take you through installing CyberDuck. There's really two downloads you need. One, you're going to need to go to the CyberDuck homepage. We don't write the CyberDuck software, so you're going to download that. It's available for Windows or for Mac. And then you have to have a one-time download of this sort of bookmark file, this connection profile. And Once you do those things and just follow the directions, it should, in my experience, take less than 5 minutes to set up. You'll see actually that here is CyberDuck, and it has connected to my data store. So as I go through here, on the left-hand side are files and folders. If I want to download a file, I can click on that file and just drag it over to where I'd like it to be. So I've dragged it over to my desktop. A new window will open, which will give me the status of the download. And I'll re-emphasize again, you may have some FASTQ files that are several dozens of gigabytes, maybe 100 gigabytes. That's getting close to your limit there. But you can actually transfer those files just by purely dragging and dropping. If I had a file that was on my local computer, so here's a folder. If I wanted to take this file, k12.fa, and I wanted to bring it over to my my CyVerse data store, it would be the same thing. I just drag it into that. And then what happens here is that you wait a few moments for that to start, and then the download will progress. And that's really it. It's purely drag and drop. There are a couple of other features where if you have a PDF, you want to preview it, or if you want to rename files or folders, but essentially it's just that easy. Within CyVerse, different folders are at different locations, as you could see here at the top. You can navigate that way, but basically that's all it takes to master CyberDuck, just dragging and dropping. In our experience, transferring something like 100 gigabytes of it's giving me an overwrite warning. I must already have that file and cancel it. In our experience, transferring something like 100 gigabytes of data from your local computer to the CyVerse data store on a good connection should be somewhere around 30 minutes. Now, you certainly want to do that. You would not want to do that in wireless, and your collection speed may vary. But the point is that it's relatively fast, so that's quite acceptable in terms of upload and download time. Okay, so that is CyberDuck. The next demo I'm going to show you is something called iCommands. If you use the command line, then this will be pretty routine. If you haven't, this is just a really short demo, but it'll give you an idea of the use of this. So back on that web page, if you remember, we were back on the CyVerse homepage in the Learning Center under Manage Data, and there's also a link to iCommands, and you can click on that link, and again, it's going to bring up the appropriate documentation. And I have actually lost it there. There we go. Okay, and with iCommands there, it goes through the things that you need. You need to actually download the iCommand tools. There they are. I will mention that the Mac and the Linux ones I know work. The Windows ones have not been updated by the people who make iCommands in some time. So I've not really seen those work, except in some cases. Really, I would say iCommands is just for Mac and Linux. Okay, but you would come down here, and there's a couple of pieces of information that you need to configure. Very similar in some way to the CyberDuck, that you have this one-time configuration step. Now I already have iCommands installed, so I'm just going to go ahead and open up a terminal. Here's one that'll be a bit easier to see. Okay, the very first time you start iCommands, you use the initiate command, or IINIT, and it will ask you for something. Since I've already done it, I'm just going to enter my CyVerse password, and nothing should happen. I should get back an empty prompt. Now, the list of commands, if you actually scroll down on the wiki, are all here. So they're very similar to what you'd expect for Linux commands like ICD or ls. All of those things really just has an I in front of it. Okay, so if I want to see the contents of my CyVerse data store, I can say ILS, and it will go ahead and list the contents. Keep in mind, as I'm running this, when I say ILS or ICD, I've already added those iCommands to my path. Please read the directions carefully if you want to do that, so that you don't have to actually execute the individual commands as they are with a /ILS. Okay, if I want to move some files, let's just do one quick demonstration. Let's say I want to get this VCF folders and put it on my desktop, so I can do an iget command. I want to do a folder, so it's going to be recursive. I want to see the progress. I want to have the time there and be verbose. And then I want to get .vcfs. I'm going to move that to my desktop. I'll hit enter. On my desktop, you can already see a VCF folder has appeared. And back in my terminal here, there will be the progress. So what I will do is go on to the next part of it. Back from the CyVerse homepage and show you the way that most people will actually go ahead and work with their data. And that is through the discovery environment. So if you, again, back on CyVerse.org, click on Discovery Environment. And there will be a link there to go ahead and launch that. I'm going to go ahead and log in. Now I've already entered my username and password because I have another window here open where I've needed to do that. You will probably just see a notification to enter your username and your password. So this is the discovery environment. It is a web interface to the bioinformatics tools, which we'll get to in a moment, but it's also an interface to data management. So if I click on this data tab here, it's going to open a window. Oops, I've clicked it twice. And if I click on my username, this is my username there. And this is the same things that I just saw a few moments ago in CyberDoc. So it's a listing of my contents of my directories. I could click on a file. Let's click on one that actually could read. So I could click on a file and preview it. Okay. I could also click on a file or a folder if I wanted to go to edit and I wanted to move that. In fact, I probably have two things selected right now. I could go to that file. I could edit it. I could rename it. I could do various things. So it's pretty intuitive and self-explanatory. But what I want to actually focus here again is on data. So if I wanted from here to upload a file, I do have the ability to do a simple upload from my desktop. If I have a URL like an FTP site, I could also import that way. But what I want to talk to you is about sharing data. So I'm going to show you a very, very simple way, two ways actually. So let's go down to a file, because this method I'm going to use only works with files. It does not work with folders. If I select a file and I want to share that file, I can go up to Share and say Create a public link. Okay, so I can come down here to this menu. Here's the one that I want to share and click Create. And it will create this URL. And if I give this URL to anyone, put it in any email, put it up on any web page, people will be able to go freely. They do not need a CyVerse account, and they'll be able to download that. So I can copy that link, and in the new tab there, I could go and I could paste in that link. First, let me go back and actually successfully copy it. There we go. And I paste that link and it will actually start a new dialog box that asks me to go ahead and download that. Okay, which I'm not going to do since I already have it. Okay, once I've created a link, then that link exists and people can use it. If I no longer want people to use that link, then I can go ahead and click this little delete icon and that link will go away. Now, if they've already downloaded it, they've downloaded it, but no one else could use that link again to download. Now, this very quick linking, as I said just a moment ago, only works with files. I would not recommend it for very large data sets, because if you try to send lots of data, invariably someone's connection will drop and then you won't be able to restart the transfer. So it's not the best way to share large files. I'm going to show you that right now. So if I have either a file or a folder, this works with anything. So I could click on something. And I can go up to Share, and this time I'm going to click Share with Collaborators. And now I can share this data with whomever I'd like to share it with. You can see I'm already sharing it with a generic account, but if I go ahead and type in the name of the person I'd like to share this with, I'll put in a colleague's name. So I start typing. They'll come up in my list with their institutional affiliation. I can select them. And then I can decide what level of access to this file. This is covered in the documentation, but essentially I could give them read access, which means that they could open the file, they could perform an analysis on the file. If I gave them write access, they could edit the file, and then if I gave them own access, they could do everything, including deleting the file. So I really have to be careful about how I share this. If I click done right now, that process would go through and they would get a notification that something has been shared with them. I'm not going to do that. Also, if I had shared something with someone and I saw them in this list because I had shared it with them, if I go back here and click that X and then click done, if it was shared with them, they would no longer have access. So assuming that they had not copied it or moved it, then you could revoke access that way. The sharing, using that, again, up in the share menu and share with collaborators, is instantaneous for the most part. So within a few seconds, really, any size data set that you have that's already within CyVerse, anyone else with a CyVerse account will actually have access to that, be able to analyze it, be able to do anything you'd like. So one thing that people will do is they'll go ahead and they'll create a folder for their lab, and then they will find their other lab mates, their collaborators, and give them the appropriate levels of access to that. When data are shared with you, they appear in this left-hand side under Shared with Me. If you're looking there, you'll see several names because there are things that are shared with everybody, but essentially that's where they go. Community data is repositories from different projects that are public in most cases. Trash is self-explanatory. Favorites, if you have files or folders that you're frequently going to use, you can actually favorite them rather than navigating all the time to go look at them. The last thing I'll mention here is that also if you are searching, so at any time you can go up to this box and type in a name, a species name, a file name, whatever you think, and you can also search. But the other really advantageous thing about the data store is that the data store is really built not just for the files themselves, but for the metadata that are associated with those files. So if I go and click on any particular file, if I go up to edit and I say metadata, go up to edit, metadata, and say edit metadata, I can actually add arbitrary metadata to this file. I can say, this is, I can have, who was the person who performed this experiment. I could have things like a time point, and I could say, 1 hour. This is arbitrary, but the more sophisticated real use case is perhaps to use one of these templates. So for example, if you are submitting samples to NCBI, which you can do directly through CyVerse, there are certain metadata requirements, and I could choose a template: sex, sample package number, tissue, cell line, subtype, cell type, disease, stage. All of those things are part of metadata. Many times we will actually have many files we want to upload, and you may have a spreadsheet of metadata. You can do that bulk metadata. But once you have the metadata, then if I go to search and actually choose this sort of smart funnel here, I can actually search in a far more sophisticated way. So I can find everything that has the attribute genus, and then the value for genus is Dioscorea, and then search. Now I can be very specific about that particular search and pull up any and all files that meet those queries. In fact, I can create a smart folder or filter where data are automatically going to appear in a folder or in a location that match the context of the search. So this is taking a moment to search. I don't even remember if I actually have any metadata that matches those criteria, but you get the idea that you can actually be quite sophisticated with your search terms there. Okay, so that is a little bit of an introduction to the data storage. So what you should be able to do, assuming that you really were able to follow that, is you should be able to use CyberDuck and think about, let me upload FASTQ files, let me upload images. You should be able to use the discovery environment, say I want a couple new folders. I want to assign permission to those folders for various people to use them. I want to be able to share these folders or particular files within these folders. So you're at the beginning of being able to get started. Of course, there's more to learn, but that should hopefully give you an idea of it's relatively straightforward to move your data into CyVerse. So we're going to move on to the next part, which is how to analyze it. In this case, we're going to be focusing on the discovery environment. So just a couple of orientation slides take us through that. So the discovery environment, as we just sort of already gotten a little bit of a preview, is a platform. And it can run almost any type of bioinformatics application. Most bioinformatics applications are run at the command line, but you don't need the command line in order to use the discovery environment. It's integrated with data and high performance, and as I said, you can add your own applications. Now, we've already covered the data window, uploading and downloading files, sharing. We're going to talk about apps, and we're also going to talk about analyses where you can monitor the job status. So we'll get into that. In this demo, we're actually going to do a really short analysis. We're going to take some unaligned DNA sequences that are in FASTA format and just create a multiple alignment. So we're going to view the data, we're going to launch a job, and we'll monitor the progress. Okay, so here I am back on the discovery environment. And let me go to data first because I want to actually show you where those data live within the discovery environment, at least the sample data that we're going to use. So I'm going to go to community data. We're going to look at training. There's one called DE walkthrough. And I think we're going to use this DE sample plant. So if I click on that one, I'll see a whole bunch of unaligned sequences in FASTA format. Okay, so how do I align them? Well, let me close the data window. I know it's there now. And go ahead and click on Apps. Okay. So this is the Apps window, or really the Apps catalog, and it's a listing of several hundred bioinformatics applications. So right now there's about 420 of them. If you scroll down this list, in fact, let me go to Public Apps. The first part is Workspace, which are things that I'm working on that may be in development. But if you go to Public Apps, you can scroll through. You may or may not know what those apps are or what they do. So you can click on the Info tab or the Info button there, and you'll get a little window that gives you a little bit of a description of what that application is. In fact, that was an HPC app. Yeah, there it goes. It tells you a little bit about it, who installed it, where it is. There's some information about the actual files involved. And there's often a link to the user manual so you can find out about that. The applications may have, let me close that, applications may have a rating, which may or may not, you can choose it yourself whether the application is working for you or not. But I'm going to go ahead and search the apps for an app called Muscle, because that's the one that we're going to use in order to just do this quick demo. And when the applications come up, one of the things is that there may sometimes be one or two versions of them. And often there is some explanation for it, but often it's different versions. So we try not to delete versions, certainly, that people will need to come back to for reproducibility. Sometimes an application will be disabled because the computing system that runs that app may not be available. Those will appear in red, and you can contact support if you have an issue that you need access to one of them. But I'm going to go ahead and click on this one because it's Start. It's the one that I know is appropriate here. And when I click on, actually, and I did that a little quickly, I click on the name of the application. It opens this little window. And all the applications, no matter how sophisticated, essentially run from this same interface. So I have a name for my analysis, and I'm going to go give that an underscore demo. I can say comments, this is for the May 2016 webinar. We encourage this type of documentation everywhere. Make sure you go ahead and give comments to things. This little green arrow you won't have because there's no grammatical errors there. It's just my computer asking me to check the grammar of things. Okay, so that it should look more like that. You can choose where the output goes, and by default the output will go to your analyses folder. But again, if you have a lab folder that everybody shares, you can go ahead and default to that one. That would be up here in your preferences, where you could choose what the default is for that, including whether you want email notifications about when analyses finish. Okay, now I call this kind of like an accordion menu. If you click on this next section of the menu, that's the next thing you have to do in the app. This is required since it has a red asterisk and it's asking me to browse for the data. I'll browse through it. It's going to open to the last place where this is open, which is actually where I want. I'm going to go ahead and select DE sample plants and then say OK. Okay, so that's correct. I have one other mandatory requirement I've got to choose, and we know that that's DNA sequence. Okay, so at this point I can go ahead and click Launch Analysis. And I'll just give it a moment. Once the analysis is launched, I get a notification at the top there. And then I can actually click on this analysis menu, and then I actually have a little bit of a history. I'll also get periodic notifications down here or up here about what's happening. So this analysis window, which is opening, is a history of all the analyses that I have been doing recently. It tells me when they started, when they finished, what the status is. The status could be submitted, could be running, could be completed, in some cases, unfortunately, be failed, but you can see what's going on. Now, if I want to cancel an analysis, I can go up to select that analysis and I can go ahead and cancel it. I could also, as we're going to do the second, click on View Parameters, because I want to see the details of that job. And in fact, it's quite useful because not only will I be able to see the parameters that I selected, like, this was a DNA sequence, it was this particular FASTA file, but there's links to the actual files. So imagine that my file names were not really human readable, or they were very long alphanumeric strings. I could actually click directly and see the input file. So it's quite useful. You can even save this and export it as a simple text file. If I click on a job that was more, had a lot more options, so let me go through and select Top Hat and go ahead and say view the parameters for that. And there's a lot more parameters. Okay. The other thing too is that if I go here and I go up to analyses and I click relaunch, I will actually be able to launch a job without inputting all of those things all over again. So the input data is the same input data I just used in that historical job. The reference genomes selected, all the analyses options are just what they were. So if I need to relaunch a job because I really need to reset one parameter, I don't need to go back and re-enter a lot of different things. Okay, so I'm going to close this. I'm going to go ahead and click refresh here to see what the status of my job is. I know actually that there are some workshops going on today, so things may take a little bit longer to complete than originally, but let's see that it's running. And we'll come back to it, or we'll go to a completed job. Oh, it is completed. So when the job status is completed, and notice I had to click refresh because I have this window open, it doesn't automatically refresh. I'm going to go ahead and click on the name of the job, and then it will open a data window. And that data window will be opened directly to the output to this particular job. And there they are. And if I click on that, I can preview the FODLIP interleaved result. Okay. So really, that is a quick summary of what the discovery environment is. So you kind of hopefully have the sense that you go to the discovery environment and you can click on data and you can see your files, you know how to share them, you can move things around. Then if you need to use an application, you can go to the apps and you can click on an app and actually load in your files and run it in a point and click interface. Some features I did not show you was that you also have the ability from the apps menu to build your own application or to build a simple workflow. There wasn't time for that, but those are some things that you can actually go through the documentation or come to one of our workshops and learn more about. Finally, you also should have seen that in the Analyses tab or panel, you're able to see the history of jobs that you've previously done. So that's your digital lab notebook in that sense. And you can also see the status of a job that's in progress. You can relaunch a job. You can export the parameters. So it's really something that's quite useful for a lot of biologists. It makes things far more simple, especially if you're not used to running things in the command line, but you don't want to sacrifice the documentation and all the other value adds that come with the way that we've been able to do things. Okay, so I have time for one last demo, and that's going to be on the cloud, but first, again, a few slides. Okay, so the Atmosphere Cloud. As I mentioned, the Atmosphere Cloud is one-click access to several hundred virtual machine images. You can customize your setup, your software, the machine itself, and it's again integrated with data and computing resources. However, many of us may not know what cloud computing is, so let me just give you a little bit of example. Again, it's not some impenetrable thing in a field that maybe we're not computer scientists. It's actually based in a lot of the technologies that we already use. So let me give you an analogy, because there's two pieces or two things I need to define. One is the term image, and the other term is the term instance. So first I'm going to start with image. Let's take a scenario. You've bought a new computer. So you need to take everything from the old computer and you need to put that onto the new computer. So you'll take those documents, files, movies, whatever it is, and put it onto an external hard drive in many cases. Now, there is actually another type of file that you could create from your original system, and this file is called an image file. And an image is really a backup, a complete backup of everything, not just individual files and folders, but your operating system, everything down to the color and the background of your desktop. An image file is a faithful copy of all of those parameter settings, operating systems, and it also would include individual data files. Okay, that's an image. Now, if you take documents off of a hard drive, that external hard drive, they're still documents. But if you take an image file, you can actually completely clone both the files, data, operating system, everything onto that new computer. Okay, so that is an image. That's the type of file I mean when I talk about an image file. The second piece of vocabulary is something called an instance. So in the CyVerse cloud, there are CPUs, there's memory, there's disk space, physical resources. And when you use CyVerse, you're asking us on the cloud here to be able to take some of those for yourself. So you'll tell us what you need. And if you're going to build this sort of what we call a virtual computer or virtual machine or cloud computing, you're going to not only need the physical resources, but also an image, the operating system to run it. But once you have both of them, you can put those together, and then you have what we call a virtual machine. An Atmosphere instance is called a virtual machine. And that's a computer that will reside at an IP address, and you can connect to it. Okay? So I'm going to show you that. Let me just also cover one or two more basics. In terms of the images, you can choose an existing one that we have. So we've already have an operating system. We may have some files on there, some software. You can also request that your instance be powered by a number of CPUs, a number of RAM, up to 16 cores and 128 gigabytes. In order to access this virtual machine, you can do that via the shell or the terminal, or we're going to use in the demo, well, we'll use both, I think, something called VNC Viewer, which is going to allow you to see the desktop. And when you have an image, you can customize it, you can share it with selected users, or you can make it entirely public. There are lots of reasons to use this. If you have software that is monopolizing your local laptop or your desktop and you want to run it somewhere else easily, that's one way. If you have bioinformatics software that has its own graphical interface, it won't run in the discovery environment, but you can run it on Atmosphere. You will have sudo access, which means that you have full administrative rights to your Atmosphere virtual machine. You can install whatever you like. If you have lots of dependencies, you won't have to worry about that. It's wonderful for education and for the laboratory because you can have a uniform setup so everybody has the same software, has the same tools, everything is uniform, and that's excellent. Especially, I use it personally in the teaching scenario. And again, if you have software and you'd like to make that available, we support the hosting of open source software, and you can release Atmosphere images that contain that software and distribute it. Okay, so in this demo, we're just going to show you how to connect. If you're using Windows, Mac, or Linux, you could use VNC Viewer. The Windows doesn't have a native bash shell, so you use something like PuTTY, and Mac and Linux do, so you could just use the terminal. I'll demonstrate all of these things. Again, when you have a chance, you can follow along via the documentation. Okay, so I am going to go back here, and I believe if you go to the CyVerse homepage, there'll be a link here to Atmosphere, and you can go ahead and launch Atmosphere. Now, when you go here, there's a listing of various images. You could search if you're interested in RNA-seq. It would come back with things that are installed with various types of RNA-seq tools. I'm going to go ahead and just click on that login so that I'm really actually inside. And I'm at the dashboard. And I get a little bit of a preview of what I'm using. And I'm going to go ahead and go up here to my projects because I already have one running. So if I go into this one, somehow I launched it. So I will show you how you actually launch one of these. If you click New and you say you want New Instance, first you need to select, well, which one of these is going to be your new instance. So I'll choose one. Then you can choose the size. Okay, that depends on whether you're in the workshop cloud or most people will be in the regular cloud. You can choose the size. Then you can click Launch the Instance, and it will start to launch. That will take about 10 to 15 minutes. So it's not instant, but it will take a few minutes. And when that instance shows up as green and it has an IP address, you can actually connect to it. I'm going to go ahead and copy this IP address. And I've already downloaded VNC Viewer on my computer. I have to paste that IP address in, and you also need to enter the colon and the number 1. I'll click Connect. Say yes, go ahead and connect. I'll use my CyVerse username and password. And that's it. I'm connected to a virtual machine. It's asking me to upgrade, which I don't need to. And I could use this as I would any other computer. I could open up RStudio and start visualizing some R graphs. I could, let me close that once it's started. I could go up to, don't save, go up to, open up the internet if I find Firefox and browse the web. Anything that you could do with your own desktop or your own laptop, you can do on your virtual machine. And if I wanted to, let me close that there, connect to this via the terminal, I can open up the shell. I could do SSH, my CyVerse username at that IP address, say yes to go ahead and connect to it, enter my CyVerse password, and I'm connected to that Atmosphere, which is right there. If I want to, I can go ahead and say, let's create a folder just to sort of emphasize that. Okay, and there's my folder on my remote computer. So this is really a general purpose tool because you can connect and at will, if you need a computer and you want to have access to that, you simply load up the right operating system that has on one of those images. Choose one that either already has those software installed, or you can actually find what we call base images that don't have very much outside of the operating system installed, and then you can install as much software, put test data, whatever it is, and then you go back and you click Image again to actually store that, and you'd actually be able to reuse that at any time. In fact, let me go back to the dashboard to show you just a few more features before we come to the end of this webinar here. So back here on the dashboard, if I look at my instance, if I click on that one, got a couple options. One thing that's important, let me click on the name of the instance itself. One thing that's important, okay, here's my instance history, is that as I'm using it, I'm actually spending time and I'm spending an allocation. If you noticed earlier when we launched things, if I actually go to my dashboard, I have every month a certain number of CPUs I'm allowed to use. I have a certain number of memory and just my allocation. So as I have things running, that allocation is going to steadily reach the point where I don't have anything, or I'm not allowed to run anything else. Now, if I get to that point while I'm working on a project, what I'll need to do is I'll need to pause. I'll suspend or stop my instance to sort of stop that allocation from being spent. In fact, we really need to stop it. If I'm not using it, I can also pause it, but I can stop as well. And if I am running over my allocation, it will automatically be stopped, but that's okay. You can get more resources. It's not in the options here, but it's actually, I think when I go back there, if I need more resources once my instance has actually stopped, I can actually get some help. And I can go up, there is a little bit of a feedback form that you would go back to. In fact, when you go and click on to launch a new instance the very first time, you also get that sort of place to sort of request more resources. So you'll always have the ability to say, I need a bigger allocation, and we'll try to help with that. The key thing, though, is that when you're done using this, it is disposable. So you would select that instance, and you can click on this X here to delete it. The really important thing is that once you delete an instance, all the data that's on there is lost. So you must transfer that data. What do I mean by that? Well, this instance has no connection to my CyVerse data store. So the very first time I start it, I'll have to connect it, and that's described in documentation. I'll bring in the data, but then I could take those data and put them back into my CyVerse data store. So read the documentation and understand how that works. And when you're done, so as not to waste resources, you'll go back and you'll delete the instance. Okay? But that's a quick run through of Atmosphere. It's super useful, and it really gives you a lot of flexibility. So the last few slides here, as we reach the hour, where to go from here. Number one, come back to the Learning Center on the CyVerse homepage, and you'll see the Getting Started Guide, tutorials, videos, and documentation, as well as upcoming events. I want to also thank, of course, these are just the executive team across the 3 of the sites who lead us in the project. And I'll emphasize, going back to the CyVerse homepage, because one of the important tools for anybody who needs help on CyVerse is if you go, it's in the Learning Center, but it's also actually in several places. If you come back to the Learning Center, you'll also find from there the CyVerse forum, or Ask CyVerse. So this is the user forum, and you can actually post your questions there. In fact, a lot of times your question may have already been answered, and so you can get help, and the entire community can see questions and respond to them. Of course, you can also email CyVerse support, and they'd be more than happy to help you find out if there's any particular thing that they can do to walk you through a challenge that you're facing in using the tools. So that's it for this 1-hour program. I'll give a moment for anyone to sort of chat any remaining questions. We didn't have that many this time, but if you do have questions, please go ahead and chat those below. For everyone else, again, besides the video recording, there is a little bit of feedback that we're asking from you, so when the program ends, we'll send you straight to the survey. As a reemphasis or reminder, if you have questions, need support, need help, please go to that link, and you'll find it in multiple places on the CyVerse homepage for the user forum. And then if you need additional help, you can also go to email support. May even have my email. But we look forward perhaps to meeting you in person. We really appreciate our users. They make the project exist, and we look forward to helping you accomplish your research and your education objectives.